I'm Matthew Eby, Regional Manager of Support Services out of Christchurch, and very happy to bring you our third installment of our winter-spring webinar series, and very happily joined by someone else in Christchurch. So that's Margreet Vissers, and just a little bit of an intro here is that she actually trained as a biochemist, graduating with a PhD in '86. She had a postdoc position at the University of Michigan with studies in oxidative stress and autoimmune diseases. She's currently research professor at the University of Otago here in Christchurch, and co-director and principal investigator at the Center of Free Radical Research Group. Professor Vissers has she's published extensively in scientific literature, over 100 peer-reviewed papers, and has received an Excellence in Research Medal from the University of Otago, Christchurch, in recognition of long-term high-level research achievement. She's had an established international reputation for research work and is regularly invited as a plenary speaker at national and international meetings. She maintains a high public profile for her work on vitamin C, cancer, and in general health. So all that said, I know I'm excited to hear more about this topic, and the topic for everyone out there, if you haven't seen it, it's new data and insight from vitamin C studies, personalized medicine in the treatment of acute myeloid leukemia. So with that, I'll hand it over to you, Margreet.
Thank you. Thanks, Matthew, and thanks so much for the invitation. I can't see who I'm talking to, but I believe that there are people out there. So thank you to the people listening for taking the time to listen to my story. So I'm
Margreet, sorry, one last thing. I'll just say one last thing to everybody that's kind of listening. Hey, if you guys just throughout, I encourage you, even if you're jotting things down, but you can put questions directly into the Q&A box right at the bottom of your screen, and then at the end of it, she has a chance to answer some of these questions.
Yeah.
Perfect.
Yes, and please do that as I go, because some of it does get a little technical, and I don't want to lose you along the way. So we've been interested in vitamin C for a long time because of its antioxidant properties. About 2000, I became more interested in its other functions because we started seeing it doing different things. And so it was that kind of ongoing interest that has eventually led us to the myeloid, or to the leukemia and blood cancer stories. And so initially I'm going to share the story, the case study with you that brought us to where we are now, and the sort of insights that has. So in 2014, I saw this item on the TV1 News about a policeman from Whangarei. His name is Anton. And I'm just saying here before we start that that I use the patient's name in this and his story at his request, and also at the request of his wife. They have requested that I not anonymize his story, and so she's, his wife has said, please use Anton's name. So I'm doing this with their blessing, so that I'm not breaking any privacy etiquette here. So in 2014, there was this item on the news of a policeman from Whangarei who had been diagnosed with acute myeloid leukemia, which had not responded to chemotherapy, and he had been discharged from Auckland Hospital, having undergone 2 rounds of chemotherapy that had failed, and was told that he only had weeks to live. And so he went home to start planning his funeral. One of his sons said to him, happened to say, "Dad, why don't you try vitamin C? I've heard about it at school." And just down the road from where Anton lived, there happened to be a doctor who administered vitamin C infusions to people, who runs a health clinic just down the road. And so he went along to this clinic. He thought, well, yes, I will give it a go. And within 2 days, his wife says, he was starting to feel better. 2 weeks after he'd had, I think, 4 or 5 infusions, he thought something has changed. And after a month, he went back to his doctors for another appointment, and they said, he bounded in off the golf course, and they said, well, I don't know what you're doing, but something's going on. And they took a bone marrow biopsy from him and found him to be in complete remission. So Anton, I met Anton. At that point, I was interested in that, and I thought, I think we have an idea what may be going on with this patient. Maybe he's in a special group of myeloid leukemia patients that we think may respond. So in 2015, I heard that Anton was going walking through the North Island. He actually walked from Whangarei to Wellington to promote the use of vitamin C for cancer patients and the availability of that. And there are pictures of him on that walk. As you can see, he's very healthy, hale and hearty. And I went and walked with him on one of these days because he had backed us in a story about our work. And I hadn't met him, and I thought, I need to meet this man and go walk one day of his pilgrimage with him. So we spent a whole day together, just he and I did manage to keep up with him just, and he put up with me for a day, but we had great conversation and became actually, after that, quite good friends. And I said during that walk, you know, Anton, what I would really like is that I'd love to get hold of some of your cancer cells, but you don't have any, because we have an inkling as to what may be going on for you. But, and so we kind of kept in touch, and it was a while later that I heard back from him. So this is just a picture of his, can you see the mouse on the screen? Yeah? Is the mouse sharp? Yeah. So this is his bone marrow at diagnosis. And so if you've seen a myeloid leukemia bone marrow, all the cells all look the same. They're all these little purple dots, and you can see they all look very similar. Bone marrow should not look like that. Bone marrow should look very diverse, in fact, like this bone marrow here. So post his chemotherapy, there was no change in his bone marrow. Post the second chemotherapy, some of the fat cells, that's these big empty spaces, had come back, but all the cells still remained the same, and there were these large areas of dead cells. So at this point, this is the point at which he was sent home. 1 month later, when they did the next bone marrow biopsy, he had all of these normal looking blood cells in his bone marrow. And at this point, he was declared to be in complete remission. So when we look at his timeline, so that was, he was diagnosed in 2013, and some months later, he started the vitamin C treatment. Over the period of the next 3 years, he slowly decreased the frequency of his vitamin C dosing. So I met him in 2015. And at that point, you know, I said, are you still doing the high dose vitamin C? And he's like, well, I'm actually too scared to stop, which is perfectly understandable. But we're just dosing it down to see how low we can get with it. And so he was decreasing the frequency of that dosing. In 2016, I received a text from him to say, you know, how you were looking for some of my cancer cells? Well, I think I have some. And he had had a respiratory illness and had relapsed. And following the relapse, he went back to Auckland for further chemotherapy treatment, because they did suspect that maybe the chemotherapy had worked on a delayed basis. chemotherapy didn't do anything. He went home, he went back onto the vitamin C therapy, kind of worked for a little while. He got well enough to return to playing golf, but after about 8 months, the cancer cells never went away the next time around. And he died, unfortunately, and very sadly, at the beginning of 2017. But in this period here, he had said to his doctor at the time of relapse, this does remind me that Professor Vissers would also like to analyze some of my cells for DNA markers. And even though I said to him, you know, really, you need to be looking after yourself, he did arrange for a bone marrow biopsy to be sent to us. We stored this at this point. I wasn't quite sure exactly what I wanted to do with it. We didn't have any staff member or any person. I thought we have one sample. We need to be very careful with this sample. We froze it away. And in 2017, we received some funding from the Bone Marrow Cancer Research Trust here, who approached us and said, did we have a project that we would like funded? I said, I have a really good project. And we assigned a young postdoc to this project to figure out what should we do with our one sample and how would we get the best information about the patient. And so over the period of the next 8 or 9 months, we had our one sample, we extracted DNA from that sample, we also looked at some of the cells, and in collaboration with his initial clinician, who was now working in Dunedin Hospital, with the leukemia people in Auckland Hospital where he was treated, and with the GP who had given him the vitamin C therapy, we put together all the case notes of Anton's case and did the DNA sequencing on his particular leukemia. What we found was actually a confirmation of what our theory had been. And so we found in his cancer cells that there were a number of proteins that are mutated. So cancer occurs when something goes wrong with the genetic information in a cell, and we found mutations in this protein called TET2 and also in this protein called WT1. Now, both of these are really interesting with respect to the vitamin C function, because this is a protein that needs vitamin C to work, and WT1 needs TET2 to work. So both of these proteins have a requirement for vitamin C in order to be maximally functional. So I want you to just remember that, because we're going to come back to these genes. So in this patient, so this one patient who had shown a remarkable and unexpected clinical response to high-dose vitamin C, we had matched that up with his cancer being particularly sensitive to vitamin C supply. And so I'm going to leave his story there for now and just go to talking about what happens and what the importance of this is. So this is very important for this one case, but how important is this for all cases of leukemia? And how does this information help us to understand what's going on? So we have Anton's one case. We also know this information that we have is on the back of what we know about normal, healthy bone marrow and blood cell development. Now, in our bone marrow generates all the blood cells that we have, and it does this in the marrow itself. So in the marrow, we have what we call hematopoietic stem cells. So these are the cells that can differentiate and become all our mature blood cells. And so there's a kind of a parent cell, then there's a daughter stem cell that makes all the white blood cells and the red cells, well, not all the white blood cells, the anti-infectious white blood cells, or the inflammatory white blood cells, and platelets and red blood cells, and they are called myeloid progenitor cells. Then the other branch that occurs makes lymphoid progenitor cells. And so you get this whole diversity in the bone marrow, and this is where you see a normal bone marrow, where you can see all these cell types present in their mature and immature forms. So you see like little cells like that are immature red blood cells. Here are some eosinophils, so that's these cells here. There are neutrophils, that are these cells here, and so they're all present. So the entire array of cells should be present in a normal bone marrow. So that is how we expect the normal bone marrow to be. When a blood cancer develops, or when any cancer develops, the cells acquire a number of mutations. So a cancer won't develop if a cell requires one mutation. So normally there are pre-cancerous conditions and then a full-blown cancer. So this is why cancer is worse when we get older. So over the course of our lives, or due to an environmental stress or environmental toxins that are cancer causing, the first cell will just acquire one mutation. Then when that cell acquires another mutation, it starts to grow a little bit abnormally, not too badly. When it gets a third mutation, it gets a little bit more abnormal. When it gets a final, what we call a final driver mutation, that cell then becomes a malignant cell and will result in tumor growth. So this is what happens in a solid tumor. Same process happens in the bone marrow as well. But the cells that get affected in the bone marrow are the stem cells. So if the stem cell acquires a mutation, and then these next progenitor cells, so these next cells that differentiate to become other cells, when they acquire more mutations, they stop being able to differentiate. So you get then, instead of there being very few of these cells and lots of these cells just start to grow, and they don't differentiate and become a different cell. So depending on where the mutations are will depend on what sort of blood cancer you have. So if the myeloid cells differentiate, you get myeloid leukemia, and your bone marrow will fill up with all of these cells like we see here. If you get the mutations here, then you'll either get immature forms of these cells, or you'll get too many of these cells. So these cells are not functional, so they don't do the job that the mature cells do. So your bone marrow at that point loses its function, and you lose all the blood cells you're supposed to have. And so that brings on all the effects of leukemias, which is anemia and inability to fight infections and all the problems associated with leukemia. Now, this process of differentiation is a different process than just the process of growing and dividing yourself. And so these cells have to become a different kind of cell. So that process requires a process that we call epigenetics. So it's like genetics, but it's a modification of your genetic code. So you may not be familiar with the concept of epigenetics, but let me try to explain that by giving the example of all the cells in our bodies. So we all start from a fused egg and sperm that becomes a single cell that has all the DNA that you will ever need, or that exists in all the cells in your body. So that initial set of DNA then ends up in every other cell type, all of it. But these cells, nerve cells, do not look like muscle cells, do not look like bone cells, do not look like fat cells. So something has to happen to the DNA to make sure that only the genes that you want to read are read. So it's like having closed sections of your library and only having some books available for different populations. So that process of switching genes on and off is called epigenetics. And there are enzymes that do that. So this is what happens in your body. It also happens in your bone marrow, the same process. So this hematopoietic stem cell has the same DNA that is present in all these mature cells. But in the process of this cell here turning into these cells, it's turned off genes or turned on genes that just these cells require. And that process is called epigenetics. Now, because in leukemias, these cells don't do that differentiation, a very common cause of leukemia is that there are mutations in the epigenetic enzymes. So the enzymes that are responsible for turning the genes on and off, they're the enzymes that are often mutated in leukemias. And I've got examples of them here. So I've just given their abbreviated names, but this particular gene codes for a protein that is mutated in 25% of leukemias. TET2 is mutated in 10%. These two proteins are mutated in another 20%, and WT1 is mutated in another 20%. These three never exist in the same cancer, and so this adds up to about 40% of all leukemias. So the effect of this is loss of normal cell differentiation. So then we get these cells staying as these stem cells and crowding out the bone marrow. And that is the basis of leukemias. And so what we also see is that there are clones of these cells. So some of these cells will have one of the mutations, and then there'll be another clone that has another one. And so they don't always exist in the same cell. So we can get clones of the leukemia, some of which will be treated with one drug and some with another. So what does this all have to do with vitamin C? And so this is where we have become interested. And so what we know about vitamin C is that it supports many enzyme activities. So it's good as an antioxidant and it's good in our skin as that, but one of its main functions is to keep these enzymes active. And so it's a proponent of these enzyme activities, and there's a number of these in the body. And the one that we're most familiar with, and the one that people have heard about because it's been known about since the 1960s, is the one that makes collagen. So collagen is the protein in our skin. If you go to any chemist shop, you will see people selling skin products that have vitamin C in them. So where people thinking you can rub it on and that will help. You should eat it, that will help. But that's another story. But we've known about this enzyme for a long time. We now know that there are many enzymes in this family that also require vitamin C. So the same as this enzyme, there are enzymes that make hormones, there are enzymes that make energy producing molecules, there are enzymes that control stress responses, and there are these enzymes that control epigenetics. So these have been known about for about 10 years now. And it was the discovery of these enzymes that made me think that a mutation in one of these enzymes could be what is driving Anton Curia's leukemia. So the link between that, between epigenetic enzymes, vitamin C, and myeloid leukemia are these histone DNA and histone demethylases. So they're a group of enzymes, there are about 20 of them, that are able to regulate the on-off switches. So there are a number of enzymes that are responsible for this, but there's a whole group of these enzymes that all need vitamin C to work. So if you don't have enough vitamin C, then these enzymes won't work very well. So, and we know that epigenetic enzymes are commonly mutated in cancer. So of those three that I mentioned, so TET2, this one here, these IDH1 and 2, and this enzyme here, WT1, they all need vitamin C for optimal activity, and they all need it. So vitamin C feeds into maintaining these activities because they all require TET2. So TET2 is the common factor here. So even when it's not mutated, these enzymes still need it to work. And so we have a whole mechanism of pathways in which TET2 is central to maintaining normal bone marrow function. And so in normal conditions, here's the enzyme here, and here it is stuck on a piece of DNA, and it works together with WT1 to remove some of these marks and switch on gene expression. It also needs these enzymes here to create this product here that make it work. So when everything is fine, there are no mutations in the cell, all of these things work, and you have normal stem cell, normal hematopoietic stem cell function. In myeloid leukemias, if you get mutations in this enzyme here, then you get another product here that stops TET2 from being active. So the TET2 is not active, and you're not getting the right products, and your epigenetics is disturbed. If you have mutations in TET2 itself, it doesn't work at all, and so or only half of it's working, so you'll have one gene mutated and one gene not, so you'll have a lesser amount of this, and you're not getting your DNA changes. If you have mutations in WT1, then you've got everything else present. You've got active TET2, but the WT1, which is also needed to cause for this gene expression, can't bind to the DNA and also can't recruit TET2. So you have three scenarios in which TET2 activity is paramount, and we need ascorbate or vitamin C to make that TET2 work. So if we come back to the mutations that we found in Anton, we found 2 of those. And so we had predicted that we may only find 1. We had expected he will probably have a TET2 mutation. So he actually had a TET2 mutation, but he also had a WT1 mutation. And we found that these 2 mutations were present in separate clones. So you had one clone of the cancer that had a TET2 mutation and one clone that had a WT1 mutation. And both of those clones were removed when he went on to the vitamin C therapy. So we could not detect any mutations or any mutated cancer stem cells while he was on the vitamin C treatment. And that was curious to us. At the time, we wondered whether it may have removed the cancer altogether. When he relapsed, only one of those clones came back. The WT1 clone never came back at all. And it's possible that that clone was completely deleted by the vitamin C therapy. The TET2 mutation did come back, as did and it had acquired further mutations. So what we realized with that is where we had thought that vitamin C therapy may be good for those patients that have a TET2 mutation, which is about 10% of patients, we realize that actually everything in this pathway could be responsive to vitamin C therapy. And so anyone with mutations in WT1, IDH1, 2, or TET2 could be vitamin C could be beneficial for this group of patients. Now, altogether, that amounts to 40% of myeloid leukemia patients. So suddenly, we're becoming much more interested in looking at what patients are responsive to this. Since this time, we've discovered that there are also other mutations that involve TET2. So we think it may actually be a bigger group than 40%. So that could be up to half of patients with acute myeloid leukemia could benefit to some extent from vitamin C therapy. What we know from Anton's case now is that because his cancer came back and we were able to sequence the relapse cancer, we realized that that whole time in remission, the cancer was still there because the same cancer came back. When it came back, when it was gone, what the vitamin C was doing was keeping those cells functioning normally. So they still carried all the mutations, but instead of the TET2 activity being decreased in those cells and causing the changes, the cancer changes, vitamin C was controlling the cancer and making it behave like a normal cell. So his cells were differentiating and turning into normal blood cells, and everything appeared normal. And so we didn't know at the time that that was the scenario. Now we know. We would manage patients differently with respect to treatment in that window of opportunity when the vitamin C is controlling the cancer. So that opens a huge therapeutic window that we think going forward will be an enormous advantage when we're looking at combining vitamin C therapy with other treatments to ensure a better outcome. Because currently when cancer cells, they don't want to respond to the chemotherapy, but if they're normal cells, they're behaving like normal cells, they may well do, and so we may have a much better chance at removing the cancer completely if we combine the vitamin C therapy with other treatments. So then we come to this, what is a bit of a fraught question, and that we know that most people take in adequate vitamin C from their diet on a day-to-day basis. And so we'd say, well, don't our cells already have enough vitamin C to function? And so there are a number of questions. So is additional vitamin C needed to treat leukemia? So was it necessary to have infusions? Is normal supplementation recommended, or should we go over and above that? And how much vitamin C is enough to give a clinical response? So this is actually a very complex question. It seems simple and you'd think there'd be a simple answer, but there are no simple answers to this question. So I'm just going to digress a bit into just a little bit about vitamin C and its need, our need for it, and why we need it, and how our bodies handle it. And so it's important to understand just why we might, under some circumstances, want to give more than we might under other circumstances. So this is humans here, along with a few other species, all other primates, fruit bats, some fish, the guinea pigs, and one or two birds. We have, unlike all other plants and animals, we have lost the ability to make vitamin C. So your pets at home, your cats and dogs and do not need a vitamin C supplement in their food because they make vitamin C in their liver from glucose. So they have a number of enzymes that make vitamin C, and we lack this last enzyme that converts it to ascorbic acid. So therefore, we need to get vitamin C from our diets. That makes it a problem for us. Your cat and your dog will keep their vitamin C level at maximum as their body makes it on demand. But we are totally dependent on what we get from our food, and so what we put in our mouth. And so we all know that, you know, health-giving foods, fresh fruit and vegetables, is where we get most of our vitamin C from. So all plants make it for their own purposes, and that's good for us because and especially raw foods, because you can destroy it by cooking. So fresh fruit and vegetables is where we get most of our vitamin C. We don't need a lot normally on a day-to-day basis. So we take it in, comes in through our gut, gets absorbed into the bloodstream, and it circulates in the bloodstream depending on how much we've eaten. So if we eat the optimal amount, our bloodstream will reach this level of about 100 micromolar. The more our body's consuming it, or the less we're eating, it can drop right down. And when you're getting down to 10, you're getting symptoms of scurvy. So every time our blood passes through the kidneys, any excess that we eat or take in through a supplement is passed out into the urine. The kidneys will reabsorb anything until our plasma is 100 micromolar. We call this saturation. So that's when our bodies are saturated, and that's why if you take a lot of supplements on a daily basis, your doctor will tell you you're just making expensive urine, because essentially once your body's at this level, all your tissues are saturated. But your bloodstream is your delivery mechanism, because vitamin C in the bloodstream is not doing anything, it's just going places. So where it's going is it's going to all the cells in our body. And so our organs take up vitamin C depending on how much they need. And so what we see is, if we look in the different tissues, that the level of vitamin C that stabilizes at saturation is different for different tissues. So it's very high in the brain and the pituitary. We know it's very important in the brain for making those brain hormones. We know it's very high in the adrenal glands, where it's really important for making adrenaline. The other place that it's really high is in white blood cells, so mature blood cells, but just in the last couple of years, it's been found to be extremely high, 20 millimolar, one of the highest levels in the body, in the bone marrow stem cells. So those cells from the bone marrow that are differentiating, those cells that need that TET2 enzyme to be functioning absolutely maximally to make sure that those stem cells become those mature blood cells. So how much we take in affects our plasma concentration. So currently, this is just a dietary advice. So you will see, you know, if you go to the Ministry of Health website, they'll have something called the recommended daily intake or the recommended daily allowance. So in New Zealand, our recommended daily allowance is about 45 milligrams, which is around about here. So this is milligrams taken in per day. So from a normal diet, the health recommendation is about 200 milligrams. So these are your blood levels. So 200 milligrams, if you're eating 5+ fruit and vegetables, will get your blood up close to saturation. But you can see here that, you know, not only get down to about 400 or 500 milligrams a day when you're eating very well, that your blood is at saturation. So this is under normal conditions. So most countries have a higher recommended daily intake than New Zealand does. So our recommended daily intake is set at a level that will stop you getting scurvy. It's not set at a level that will keep you healthy. So that's important for thinking about ongoing and daily usage. So when we look at our general health, our general population, and we see, you know, how's our population faring in terms of their plasma or their blood vitamin C levels, then we see that the average is about 50 micromolar, which is about halfway to saturation. This is very typical of Western cultures. So we're not eating probably as many fruit and vegetables as we should. So our normal healthy population is about here, but you can see quite a significant proportion below that. And some, you know, it's not that hard to find people who are scurvy or close to scurvy. So these are just our well population of 50-year-olds. The situation changes, or the picture changes, when people get sick. So one of the things we know now is that when people are sick, and it pretty much doesn't matter what you're sick with, but the more acute your illness, the worse the situation is. But this is a picture or a snapshot from a university teaching hospital. So these are people, the general population. So this is blood vitamin C levels and number of patients in white here. So the normal population, you can see that they're, you know, once again, all up around that 50 and, you know, with a good distribution curve. The black lines are people being admitted to hospital. So on admission to hospital for any reason. And so you can see that people, when they're being admitted to hospital, they're coming into hospital, most of them with levels below 25, which is at risk of scurvy. Some people are well in the scurvy range. This is just a snapshot of like a whole population of unwell people. This is really well established now. So this is one study. There have been numerous studies like this. And what we know for our requirement for vitamin C is that when we're sick, our bodies are consuming much more vitamin C than they normally would. So if we're not matching that consumption with additional intake, then our bodies get depleted. And so I often use the example of, you know, we know when we get a cold that one of the things you feel like doing, and most people do, is make a lemon drink, or they go and buy some citrus fruit, or they go and actually buy some vitamin C tablets or something. And people kind of gravitate to that. And I think your body actually becomes hungry for that vitamin when you're ill. So the depletion of vitamin C when you're sick, much faster than any other vitamin. Our bodies don't store this vitamin, and you will become deplete really quickly. So how does that impact on patients with leukemias? In the last, and particularly, how does it impact on the epigenetic pattern? So in the last 2 years, 2 papers have come out where people have measured vitamin C status in patients receiving either conditioning chemotherapy or stem cell transplants. So people actually undergoing chemotherapy and then having a stem cell transplant. And so the first study came out last year in 2019, and they both show essentially the same results. So this is an American study. And you can see that when people are admitted, so they did something like 20 patients of all types of blood cancer. So they're not just the myeloid leukemia patients, but they're all the blood cancers. So these are people about to undergo the stem cell therapy. So you can see that when they're at their first measure, their levels, the average levels are about 30, and they're kind of scattered here. When they're admitted to the clinic, they've already dropped in that intervening period. During the time of treatment, so by day 14, almost all the patients are in the deficient range, and some of them are in the scurvy range. Then, as their treatment's gone on, and so they're kind of post-treatment now, they return back to normal. So we can see that not only does their level of illness impact on their vitamin C level, but so does the treatment regime. So that's the first paper. The second paper was actually from here in Christchurch, and it's with a similar group of patients. So same patients undergoing the extensive chemotherapy and stem cell transplantation. And so you can see here when they're at week 0, their levels are, there's a broad range of levels. As they progress through their therapy, their levels drop until at week 2, which is very similar to the other study, many of the patients are deficient. And whereas, and then post that treatment, as they return to normal eating and more normal functioning, their levels rise again. So what we're seeing in these patients is that just the treatment alone is impacting on their vitamin C status, and that will be impacting on how much vitamin C is available for those stem cells in their bone marrow. So this is kind of influencing our thinking around how much vitamin C should we be giving these patients, and does it matter, can we get enough into someone orally, or should we be giving it to them intravenously? And that there's quite a different kinetics around that, so the two things are not quite the same. You don't deliver quite the same amount of vitamin C if you're taking it orally than if you're taking it intravenously. So this is a curve here. So if you eat 10 grams of vitamin C, which is a lot, then your blood level basically is regulated by your kidneys, and you will never go up above that 100 micromolar. If you bypass the gut by injecting it immediately, you can, or directly into the circulation, you can transiently, so for a period of some hours, reach very high levels in the bloodstream, but that will drop over a period of hours as it's cleared from the kidney. So there's a lot of discussion about whether having that high peak available for that short amount of time actually makes it easier to deliver vitamin C to those cells in the bone marrow. We're wanting to do some studies on this. Last year, some of our colleagues in Denmark published the results of a first study that they did, where they've seen a benefit of oral vitamin C supplementation to patients with myeloid cancers, so lots of leukemias, in conjunction with azacitidine, which is an epigenetic treatment. And they've looked at the loss of vitamin C, how much does oral vitamin C actually replace the vitamin C loss. And so you can see what they did. So it was a randomized controlled trial. They had 10 people in the vitamin C group and 10 in the placebo group. And they see the same thing as I showed in the previous two papers. So this is their placebo group. And so just constant, so these people have been through cycles of treatment. They've received vitamin C in the last 2 cycles. That's the green line. So when they were recruited, 40% of them were deficient in vitamin C, 30% were marginally deficient. So many of their patients were below, are low to start with. These 4 patients here were taking a vitamin C supplement at the time of recruitment. So over the period of time, they all dropped, and many in their placebo group stayed really low till the end of their treatment. In their supplement group, they started supplementing their group here. And so you can see that for the bulk of there's a couple that are already taking a supplement, but for the others that where they have given 500 milligrams of vitamin C a day, that they've managed to restore their blood levels of those patients to close to saturation. So they've normalized the blood level just by amping up the oral intake. What they also saw, and they've got initial data on this, is that they saw that restoring those vitamin C levels affected the epigenetic changes in those leukemia cells. So just look, so their placebo group or their vitamin C group, there's a difference in this measure here, which is measuring that TET activity. So essentially they're showing more TET2 activity in their vitamin C group just from oral supplementation. They also show that their severely deficient group, so anyone who was severely deficient, had much lower measures of TET2 activity. So they're actually correlating your vitamin C status, just your plasma status, which we know you can supplement orally, with the level of this enzyme activity. They've not done a clinical response on these cells, so we don't know whether or not they've provided enough for that. So I'm just going to come back with that to Anton Curia's dosing schedule, because this is still an unanswered question. We know from him that he started on intravenous vitamin C and achieved the complete remission in a very short space of time. We also know that he was decreasing the frequency of his dosing here, and he got down to maybe once a fortnight. So he was having an infusion every other week. We don't know whether his respiratory illness that he got depleted his vitamin C levels even further. We suspect at that time he's like, well, I'd kind of backed off on the diet, wasn't eating quite so well anymore, and he got this respiratory illness and then the cancer relapsed. So we're not sure at the moment whether or not to maintain a remission like he had, you need to be very vigilant about your vitamin C levels, how much you need to give for that, just how long that would have lasted even without him getting ill. So there are a number of questions still, but it seems really interesting that it's possible that even dietary vitamin C, we may be able to achieve a result here. So it looks really interesting. It looks like we can move forward to explore which patients, these are the questions that we want to answer soon, which leukemia patients benefit from vitamin C therapy. With Anton's case, we have identified one group. We believe this may potentially be broader, given the importance of TET2 to the function of these cells. We want to figure out what doses should be used. We want to know, is high-dose therapy advantageous? We know people can tolerate the high-dose therapy well, but we don't know whether it's a necessary thing. We want to know, how does vitamin C interact with other therapies? Because we expect that it may well be really good in conjunction with some of the standard therapies. And only future clinical studies are going to address those questions. And we're definitely hoping to get on with those. So I'm going to end there and just thank the people. There's a number of people who've worked with us on this project. So Andrew Das was the postdoc who worked on determining what the mutations were in Anton's case, together with the leukemia team from Auckland Hospital. Lucy Pemberton was the clinician who worked with Anton, who was his initial clinician in Auckland, and Damien Wojcik is the clinician, or is the doctor in Whangarei, who administered the vitamin C therapy and kept really good notes. And particularly I want to thank Anton and Louise Curaya for sharing their story with us, because his generosity has brought us far. And so we're extremely grateful for the opportunity to carry out that investigation. So thank you.
Margreet, that was a great presentation. So much to take in. Can you hear me here? Yeah.
I can hear you. Yeah.
Oh, get out. And I thought it was just, it's just phenomenal, the power of one, you know, starting with Anton, yes. And how it just, you know, spawned this, your research and other research.
Well, his case is it had, you know, some people have done, some animal work had been done. His case is and remains the first in human demonstration of this actually working. And so there's a significant amount of interest in the publication that we've generated from that.
Oh, good. Now, Margreet, can you see the questions sort of in the box at the bottom?
No.
Good. Okay.
Do I need to finish sharing the screen or something to do that?
Maybe stop sharing that, because I think it'll allow me to get back.
Stop share. Okay.
Yeah, good. And yeah, okay, I think we
Q&A, I see now. Yes.
Yeah, there's a few questions. We can sort of go from the top, or if you want.
I have it.
Yeah.
It's Louise Curaya. My goodness. Kia ora, Louise. Thank you so much. I was going to let you know that I was doing this, but thank you so much. She's a Wonder Woman.
And then there's a question below. I think that was what about the plasma cells? I think that's when you were commenting on the slides early on.
Right.
Yeah.
Well, plasma cells are a differentiated lymphocyte. So we call them plasma cells. So when the mutations occur in a plasma cell, then the cells have actually gone through to that stage, but the mutations in the plasma cells, and this particularly happens with myeloma cells, the mutation then is generally not an epigenetic mutation. It's a mutation that stops those plasma cells from dying off, and so they then accumulate for another reason. So different from the blast cells that accumulate, they accumulate because they're not turning over like they should, so which is what makes them very hard to treat, because they are the ultimate survivor. They've turned themselves into the ultimate survivor cell, and so, which is why some of the new drugs for myeloma are much better, because they target those pathways, turn those cells on to dying like they normally should. So whereas chemotherapy for myeloma is very ineffective because you'll kill everything else, but these cells will survive. So that's a plasma cell.
Okay, very helpful. Hey, I'll just drop down a few and read it out for everybody. The one from Jeanette: is there any evidence that vitamin C is useful in blood cancers other than AML? Yes,
there is, and so that, you know, that's the reason that I kept that. You know, initially AML, acute myeloid leukemia, is the epigenetic pathways are well documented in that, and due to some very excellent publications from about 4 or 5 years ago. And so we know a lot about the epigenetics of those pathways, but the other blood cancers, some of the lymphocytic leukemias and chronic myeloid leukemia are also responsive in this way. It seems that the epigenetic mutations are some of the first for the cancer cells to acquire, and they will take those mutations first and then acquire other mutations that affect downstream things. So the epigenetic regulation seems to be quite common, and therefore potentially this, which is why we're interested in looking, first of all, we're interested in very targeted approach, but with the more we learn, the more we're thinking we actually just need to look in everybody, and we need to look in all the leukemias and see who responds, and then look at why they responded, because we think that this could be a lot broader than it is. Not everybody will necessarily have the components like Anton did, but we expect a range of responses, from partial responses to complete responses. And there will be some cancers that don't respond at all, just because of the mutations that they have. But interestingly, the group of potentially susceptible people is growing rather than becoming more narrow. And so we're very keen to get, kind of refining our proposed clinical study to be more inclusive of patients, and to see, well, let's see who responds, because there are so many mutations involved and so many combinations of mutations that it's quite hard to predict just from each individual case. Like, we didn't know about the WT1 involvement until we saw it. And it was only then that we went back and looked at how does WT1 work. And well, actually, look, it has a absolute requirement for TET2. And so upregulating TET2 will correct a WT1 mutation, whereas you wouldn't think it would. So, we're learning all the time. We're learning all the time. It's a very exciting time to be involved in this work.
Appreciate that response. I'm just looking, I dropped down a couple, someone, Tamsin said, just an aside, did your example patient have a stem cell transplant at any point in their treatment?
No, he did not. No, there was not a suitable donor.
Okay. Yeah. And then there's another question about the level of dosage, but I think that's still, how much does a patient need to take? But I think that's a question you're still trying to work out.
Yeah. Yeah. We're interested in that paper that showed that, you could achieve, some level of normality just with oral dosing. And also, those recent papers that have shown just how much the levels plummet in these patients. What we also know is that out of all the patients that they looked at, the myeloid leukemia patients, when they walk in the door, they were all deficient. So the myeloid leukemias in particular seem to be going through a lot of vitamin C. So it's very possible that you may need only oral dosing for some cancers, but that for others, we may need to be giving a lot more than you can manage to eat.
Okay, gotcha. Hey, there's just this other one, it might get a little technical, but it says, my understanding from your talk is that a TET2 is a tumor suppressor gene. What was the driver mutation or oncogene, and how would the vitamin C treatment affect and apparently halt the activity of this oncogene during remission?
TET2 is a tumor suppressor gene. It's also a driver gene, and WT1 is also a driver mutation. So because that depends on TET2 for activity, with all of these mutations, you will have all of our genes. We have one copy from each parent, and so you have a mutation in one of those copies. So when you have a mutated TET2, your cells still have TET2 activity, but they only have half the amount. The same with the WT1, so they'll only have half the amount. So essentially you're working on a suboptimal basis, which is why we think the vitamin C works, because it can boost the activity of that remaining TET2 that then restores normal function. So it's dependent on you having one gene mutated, which is what happens all the time. But yes, TET2 is a tumor suppressor gene, and therefore restoring its activity restores normal function. I think because you need all of the mutations in order to sustain that cancer cell population, if you restore enough normal activity, the other mutations then only put that cell into a pre-leukemic state. It's no longer a full-blown blast leukemia. And so that's why we think the cancer was still there, but it's just that those cells were so few they were undetectable.
Gotcha. Yeah, thank you for that. Hey, there's another one, Margreet. It says, can you comment on any other dietary changes about achieving or maintaining remission? For example, she says, I've heard that fasting can positively affect achieving remission.
It's hard to know. There's a lot of interactions between cancer and metabolism, and we know that your body's metabolism changes when you're fasting. I wouldn't predict that would have a major impact, but it may affect your general well-being. So it does. It's supposed to be good for us to spend some periods of time not eating.
Right.
So, but it is. So whether some dietary things could definitely help, and a lot of them help with maintaining general well-being. I think in the case of vitamin C, we're wanting to restore the loss of a specific nutrient, which may come along with a healthy diet, but cell metabolism is very complicated in cancer, and we know that cancer cells are generally driven by like a non-oxidative energy process. So they've switched over to using glucose as their main energy source and working anaerobically. And there are some thoughts that when you're fasting, you can kind of induce that switch back a little bit. So there's a lot of discussion about it, but we're not actually sure of what the mechanism is. But there are lots of interesting things going on.
Gotcha. Yeah, gotcha. Hey, there's this other one, anonymous. It just says, are there any cancer treatments that would be contraindicated that aren't kind of known with vitamin C supplementation? They wouldn't work so well if you're taking drug X with vitamin C sort of thing.
People worry about this, and it's not an unfounded worry. So vitamin C is very reactive with a lot of things, and so people worry that it might interfere with some treatments. It's hard to know. It's hard to know until you look. And so sometimes things that are predicted to happen in the laboratory setting don't actually happen like that in the body. And so we can worry about something that it's going to happen in the body, and it turns out it doesn't, because the vitamin C is managed and handled by the body in its own way. So but it's definitely worth keeping in mind that there are possible contraindications. And so that, you definitely wouldn't want to starve someone of their vitamin C, because that is really going to be a bad thing. But just in terms of how much we should give and who we should give it to, I mean, that's why we have all these questions around how much is the right dose, what are the indications together with other therapies, which therapies would it work best with? If so, how should you give it? Should you give it beforehand, or should you give it with the therapy? Or, is this a preparative kind of process that you put the cells into and then go into the other therapy? There are lots of questions, but we need to keep an open mind on it.
Okay. I appreciate that. I think we're just winding down here. There might be one or There is a question, I'm combining sort of two. If you have a comment about lipospheric vitamin C, is that sort of worth it, for someone that's having a good diet, but plus lipospheric, and there's a cost to it, of course.
Sorry, I'll just reply to Louise.
Nice.
Sorry, which one was that?
Basically a question about lipospheric vitamin C.
Oh, right, yes.
Yeah,
Yes.
sort of...
No, I think if you're a healthy person and you're eating a healthy diet with a good diet, you do not need to be taking lipospheric vitamin C. So there are much cheaper ways of getting your 200 to 400 milligrams a day. Fruit and vegetables, and that will also give you additional benefits. We maintain fruit and vegetables first, always. Food first. If you can't, some people have difficulty, getting enough from their food because of particular dietary restrictions. Normal supplement, your body cannot tell the difference between lipospheric vitamin C or vitamin C from a tablet, just a supermarket tablet. So it's very water-soluble product. Your body handles it. We've got, all our cells have receptors for taking it up. Your body can't tell where it's come from, can't tell the difference between something out of a bottle or something out of food. And so you'll essentially take up pretty much everything that from your gut that goes through. And then your body will handle it and get rid of any excess. So lipospheric vitamin C, as people do swear by it, but I'm not sure that we have yet to show that it's at any advantage. So
okay, gotcha.
Very easy to get it cheaper.
Okay, because then there is this other question from someone about they're interested in like a depot injection, an injection to deliver, to top up a sick person's levels, bypass the kidneys and deliver an oily solution.
Yeah, I mean, I'm very cautious about doing anything like that. I mean, our bodies are adapted and primed actually to take vitamin C up to meet our needs. And so anything that you put in your mouth will be taken up, without you needing to make sure it stays around longer, or, your body won't store it though. So you do need, if you're sick, you do need to have some every day. So in fact, even if you're not sick, you need to, if you have a binge for a week on fruit and vegetables and you think, great, my vitamin C levels are great, and then you go back to your previous diet, within a week your levels will have dropped back to where they were before. So it is something we need to do on a daily basis. And I don't know that that bypassing the kidneys is a good idea, because vitamin C becomes oxidized in your body. And when it's oxidized, you want to get rid of those oxidation products, and your kidneys will do that straight away. So actually we want that system to be functioning normally. So, good kidney flush out, nothing wrong with it. We actually want that to happen. Mm-hmm. So it maintains the level in our bloodstream at 100 micromolar, because that's what will get to the tissues without it getting too high for too long. So, it is, our bodies manage it really well. So.
Okay, appreciate that, and thanks for handling I'll just maybe leave this as the final one. I know our time's running thin.
So
they just
the
said,
last question, can vitamin C be administered while preparing for a bone marrow transplant? I see no reason why not, but I would talk with your doctor if you're thinking about having infusions. So.
There you go. There you go.
You certainly can't, taking a tablet is never going to upset the equilibrium. So.
Okay, well, I just want to kind of wrap things up, and another big thank you. This will be very exciting to have this sort of on our archive and recorded. And thanks for handling us, peppering you with questions on.
No. I'd like to thank everybody for listening in. I haven't been able to see you, but I'm really grateful to be able to share the story, and I'm particularly touched that Louise logged in as well. So this is her story. This is hers and Anton's story, and we're taking it forward, and wanting to make sure that it's not the last story like that. So.
So beautiful. Yeah. Hey, I'm saying this personally. I think I'll take out of this. I'll definitely be doing more fruit and vegetables ongoing. Yeah, moving forward. But hey, Margreet, seriously, yeah, we just want to thank you again. Massive thank you on behalf of patients and families listening around New Zealand, and from LVC staff. Like, we really, you put the effort and the time in.
Well, we're really excited about it, because we think that we're at the point of being able to make a real difference, and that's if we can, we have clinicians working with us who are very keen to advance the story, and the great thing about this is that it will take no time at all to get through to the clinic. There is no registration required, for adopting this into your clinical practice. So it's just a matter of demonstrating that, how to handle it, and then we're away.
Well, good. Thanks again, and thanks for joining on our webinar series. You go well, and
thank you.
All the best to everybody else out there.
Yeah. And the same everybody else too. We will beat COVID again.
And you keep up your great research. Take care for now.
All right. Thanks. Bye.
See ya.